All right, thank you all for coming on out. I know it's a rough time right after lunch. All right, so I'm going to give a kind of an insect overview. Oh, before I do that, I've included here my email address and cell number. Telemarketers already have my cell number. They got it shortly after I got the number. So I don't care who has it. All right, so starting off in the year, in the spring of 2018, we actually start a little bit farther back in the fall of 2017. In the fall of 2017, we had very little aphid pressure in wheat. Fast forwarding into the spring, Bill Sissel and I were conducting surveys of small grain insect pests. Aphid pressure in small grains was also pretty low. We saw very little barley yellow dwarf virus in 2018. We also had pretty low cereal leaf beetle pressure. About a third of the fields that we surveyed had cereal leaf beetle in them, never above our threshold, or never really nowhere close to our threshold of 25 eggs or larvae per 100 tillers. Our first cereal leaf beetle eggs were observed on April 13th in a field near Milford, and our first larvae were observed on May 11th at a wheat field near Harrington. We were conducting this survey with the goal of relating cereal leaf beetle phenology and egg hatch and egg laying with temperature, looking at Virginia Tech's degree day model. So as some of you may appreciate, the year that you make a big effort to look at a particular insect pest is the year it disappears. So you're welcome. This year, true armyworm was pretty active in southeastern Virginia, northeastern North Carolina. Dominic Reisig, North Carolina State field crop extension entomologist, wrote in his blog that true armyworm outbreaks in small grains occur on a 10 to 20 year cycle in North Carolina. This was their year. We did have a bit of true armyworm activity in the central northern part of the state between Dover and Middletown and west out to the bay. That seemed to be our armyworm hotspot. In general, outside of that area, armyworm populations were spotty in small grains. In Sussex County, they were almost non-existent. Rains that started in mid-May really favored diseases of insects. This here, some of y'all can see this. This here is what used to be an armyworm. This black, brown cadaver on the head of a wheat plant. That is an armyworm that's been infected with fungal pathogen. It's getting ready to split open and spread spores throughout the field. Now, some of our armyworm pressure came in late, especially on barley. And we need to keep in mind that our pyrethroids have long pre-harvest intervals. Mustang Max has the shortest pre-harvest interval of any pyrethroids, and it's 14 days. After 14 days, we're limited to Previthon. Previthon is a really good worm product. It's a little bit pricey, but it's a fantastic worm product. It has a 1-day pre-harvest interval. So I've got a question here. What is the role of a prophylactic insecticide for armyworm and cereal leaf beetle management? This is an open-ended question to y'all. We're dealing with an insect that is not reliable. It generally has pretty spotty populations, and prophylactic pyrethroids are an easy and common thing to do at flag leaf and flowering fungicide applications. I have not heard of any fields that hit armyworm thresholds that also got a prophylactic pyrethroid. But we also had a lot of rain. I have a hard time thinking that the residual activity on these products is giving us that control. Could be that we have a spotty pest to begin with. Could be that as those pyrethroid residues decline, they become repellent, which has been observed for other insects. So this is an open-ended question. I welcome y'all's feedback. For cereal leaf beetle, we weren't going to hit a whole lot in wheat. Just our populations were low. In the springtime, we were surveying fields for slugs. We had a couple of fields in Sussex County that had really good slug populations, and I started getting excited about doing some bait trials with them. And then it turned warm and dry at the beginning of May. It's hard to think back that far now with all the wet weather we've had. But in a couple of these fields, our slug populations just never recovered from that dry spell. And then it started raining. And some fields that couldn't get planted until after it stopped raining in June, by that point it warmed up, beans jumped out of the ground. But fields that were in between were at high risk for slug injury. And we saw that in some spots. So Virginia is doing quite a bit of slug research right now. There's a graduate student, Kirsten Brichler, with Sally Taylor's group. She's been surveying soybean fields throughout the Shenandoah Valley region. That, for Virginia, that is their slug hotspot area. It's a little cooler, a little wetter. And so she's been measuring landscape features, including previous crop, current crop, cover crop, insecticides at burn down herbicide, insecticide seed treatment, timing of that burn down herbicide, residue cover, use of row cleaners, all these different characteristics. Can anyone guess what characteristic she found is most associated with slug injury on the crop? Feel free to shout it out. I know. Burn down timing.
Early pre-plant burn down.
Okay.
That's a guess.
Anyone else? So I asked her, I was like, well, what about soil moisture? That was my guess. Insecticides. Insecticide usage was the only factor so far, as the data is being crunched and calculated, that has been associated with slug pressure. We've got several species of ground beetles. These two are our big slug predators. There are a few others that will also feed on slugs. Slugs are insensitive to pyrethroids. They're insensitive to neonicotinoid seed treatments. Our ground beetles are not. These guys are free pest control. If we can conserve them, I think we should make an effort to do that. Dr. Ivan Hiltpold on Maine campus, he's with the Entomology and Wildlife Department, and Bill Sissel were conducting slug surveys throughout the state looking for natural enemies. In some fields, about 12% of the slugs that they collected brought back to the lab turned into this about 3 days later. You put this under a microscope, and no, this is not a cracked microscope lens. All the white squiggles, those are nematodes. This year we had really good weather for entomopathogenic nematodes. We think of nematodes primarily as plant pests, but there are large numbers of nematodes that will infect insects, and apparently we have a slug-killing nematode out there too. In Europe, there is a nematode that goes after slugs that has been commercialized, mostly for the home gardener market. It's very expensive. It's a little tricky to use. Nematodes are very sensitive to dry weather, but it's been commercialized. Anything that we can do to learn more about our system and all the factors that influence slug populations, the better off we are. The more we can start looking at these things and say, hey, how can we manipulate them or work with them to maximize slug management? So that story is right at its beginning. And so we'll be keeping tabs on it. I'm pretty excited about slugs and nematodes. In Delaware, we conducted quite a few slug bait trials this spring. One of the questions I had was, how long does a bait last in a field? So we spread out some bait in a field, and we brought that bait in a week later after it had gotten 4 inches of rain. And one thing I was impressed by, this here is a deadline pellet in a puddle of water. I was impressed that they were still holding together first. We were still able to actually work with them, find those baits that we spread out a week before. And we took those baits and we put them in petri dishes with a single slug and a dandelion leaf for the slugs to feed on. Here's our untreated check. We've got quite a few holes in those leaves. We put out a deadline and another product called Ferox. And you can see no holes in the leaves. Those slugs fed on the baits and died. So that active ingredient in those two products is still effective after getting a lot of rain. The field situation with slug baits is another ball of wax completely. Scott Stewart is an extension entomologist in Tennessee. He asked the question, can we blunt a slug population early enough that we don't have a huge problem with stand loss? In soybeans, we can tolerate some stand loss because of plant compensation and neighboring plants growing larger. So what he was doing was applying baits at about 5 pounds to the acre with herbicide burn down. So I was like, hey, well, that's not a bad idea. We've got some extra bait this year. So we did the same thing. And our goal was to put out our bait about a week before planting in these fields that we had quite a decent slug population. In reality, the fields were planted a full month after we put out our baits. By that point, slug eggs had hatched in the soil. We did not get any significant reduction from our baits, nor did we see any significant reduction in the percentage of soybean stand loss. It was low. We had about 6 to 10% stand loss in this field. Maximum number of slugs that we had per square foot was approaching 3, maybe slightly over 3 slugs per foot. We also had a field in Sussex County that had been replanted because of slugs. And the farmer decided to apply a bait after he saw that the replant was also getting worked on. And so we put out these tarps to shield areas from receiving that bait so that the deadline bounced off of the tarps. Some of them we put Ferox underneath, and others we left as untreated. Between 5 days and 8 days after we put out our baits, we got a nice little bump in soybean stand. Having said that, what I don't have on here is how many row feet that's out of. That's out of 20 row feet. Is one plant every foot an acceptable stand? After about 8 days, we started seeing a decline in our stand. This is measured from the same spots in the field. And that's a combination of weather and slugs that are underneath of a really thick thatch layer. This field was eventually replanted a second time. It was replanted at the end of June. It turned very warm, very dry. Soybeans leapt out of the ground very quickly. So, how effective is a bait alone? Everyone I've talked to says that we're not going to get slugs with just a bait. There is no good field cure for slugs. Best bet is to sample before planting. A population of around 3 slugs per square foot or more puts that field at an elevated risk. And I say a risk, it doesn't mean it will have a problem, because slugs are very weather dependent. If we can, avoid a neonic seed treatment, avoid a pyrethroid early, because both of those products will work on our ground beetles, which are giving us free slug control. And then watch that stand emergence like a hawk. We don't want to be behind the 8 ball on that. Now, my last big point on this one here is we need more data on bait with and without vertical tillage. Joanne and Bill did quite a bit of studies a few years ago looking at vertical tillage. It can work on slugs pretty effectively. How well does vertical tillage alone versus a bait alone or the combination work is still kind of this hazy, open-ended question. This here is another special stand-reducing oddball that we observed in a single field earlier this year. Can anyone guess what is doing this to soybeans? There are no bad guesses. This is truly an oddball. I had to go back in 2010 and 1999 Iowa blog post to find a similar discussion of stand reduction like this. This here is a hole in the ground. We've got a soybean leaf right outside of it. These are cutworms. We had between 5 and 10 cutworms per square yard in this field. And I hope it'll be another 20 years before we see that again. Due to various factors, the grower was not able to rescue his stand from cutworms, and it had to be replanted. So by the time we started getting sick of rain, it's like the tap shut off, and it stopped raining. One of the pests that we're looking at this year, I have extension responsibilities for both field and vegetable crops. I've done some work previously on spider mites in North Carolina, so I was excited to work with spider mites in watermelons. So we've been conducting spider mite surveys. June 11th was the last day it rained for us in June in Georgetown. We picked up our first spider mite in watermelon on June 12th. So we're looking for natural enemies. And what we found were 2 species of phytoceid mites. These are tetranychid specialists. They feed on European red mite, 2-spot spider mite, carmine mite, Neocelus phallacis. It's a kind of a pear-shaped, straw-colored mite. And I'm not even going to try to pronounce that name, P. mexicanus. Both of those were identified by Monica Farfan, a postdoc working in Clemson, and Rebecca Schmidt-Jeffers, extension entomologist for vegetables at Clemson. This mite, Neocelus phallacis, it can be a significant spider mite predator in orchard crops. In field and vegetable crops, what's its role is a really good question. The fields that we found it in, I felt like it was never able to keep up with spider mites. In orchard crops, it can. There are other predatory mite species out there. Some of them, like phallacis, are commercially available. It's something that I want to start looking at moving forward. Other natural enemies that we encountered, this is probably the most important one, Stethurus. It's a very small black lady beetle. I have samples and alcohol here. If any of y'all want to see that, feel free to come on up later, and I'll hunt down my Stethurus samples. We did a miticide trial in watermelon at Carville this year. We infested our watermelons with a population of spider mites that had previously only been exposed to Spirotetramat. It's the active ingredient in Contos and Movento. It's a great aphicide, not a very good mite product. Mites are listed as suppression only. And that we got some really good mite pressure when we inoculated our plots. We gave our plots a little bit of help. Gordon probably does not appreciate my spreading spider mites across the station. He had issues with them in his tomatoes. Towards the middle and end of June, our spider mites were increasing at a rate of 10-fold a week. So my goal was to hit these spider mites closer to our action threshold of 1, 2, 3 mites per leaf. In reality, I hit them at anywhere from 50 to 150 spider mites per leaf. We had a glorious infestation, a little bit too late. I was very pleased with how all of our products worked, but especially with Monecto and Zeal. For those of you who aren't familiar with Monecto, it's a premixed product that has abamectin and chlorantraniliprole. So it picks up mites, picks up worms. Having said that, both Portal and Oberon, those are contact-only miticides. We're going out at 40 gallons per acre on a really high population of spider mite. Spider mites make a lot of webbing. That webbing shields them from a lot of our pesticides. And I was using 40 gallons per acre. If you're going to go out with these products, bump up that gallonage. We need good coverage underneath of the leaves. We had spider mites showing up in soybeans. Some of them were causing some somewhat significant looking defoliation. This here is the edge of a soybean field, extending out about 50 feet. You can see these plants are a little bit yellower. The rest of the field was fine. But that was very noticeable from the roadside. That was taken July 19th. Now, Mississippi State did quite a bit of work the last couple of years looking at defoliation on full season beans. And in their environment for full season beans, it's very difficult to get a yield response to defoliation. Here they defoliated the plants at 60% at V3 and 60% at V6, and they still did not get a yield response. For those of you all who were in David Holzhauser's talk yesterday, what was something that he stressed about defoliation for our double crops? Right. We need to keep our double crop leaf area. For full season is a lot more forgiving. So bottom line on spider mites. We can't neglect our field interiors. Spider mites will balloon on air currents. So they can show up in the middle of the field. More often than not, they're going to be on the field edge first. And that's where I go to look for spider mites. Cool temperatures, high humidity, and heavy dew is a spider mite's enemy. Spider mites are very susceptible to fungal pathogens. I tried to get in a miticide trial in soybeans this year. My mite colony came up around the beginning of August. Middle of August, I hit them. 3 days after I hit them with miticides, there wasn't a single live spider mite left in the field because of pathogens. So fungicides can limit mites and mite pathogens. Now, having said that, it's been raining since, September, beginning of September? I collected a very nice population of spider mites out of a corn maze in October. That was fun. I was a little jealous, actually. It's like, why? Why can't I get these guys in my soybean field? Neonics, they can stimulate mite reproduction. They act almost as a hormone for spider mites. So those spider mites actually kick reproduction into higher gear. The downside on that is for vegetables, we need that class of chemistry to manage other insect pests, and we need those fungicides to manage other fungal pathogens. So that puts vegetables at a disadvantage mite-wise. Once they get established in vegetables, they're much harder to dislodge with just environmental conditions. Spider mites also have very limited outbreeding. What that means is in a field setting, those spider mites they're not going to reproduce with susceptible mites from outside of that general area. So if we hit them multiple times with the same product, we're going to drive resistance, and they have limited options to incorporate susceptible traits back into their population. So, what can we do for the spider mites? Preserve our natural enemies as best as we can, limit some of our broad spectrum insecticide use, and secondly, if we have to hit spider mites this year, or if you have to hit them more than once, and oftentimes we do when they show up, especially in vegetables, rotate mode of actions. And I would say if you hit them with miticide number 1 this year, hit them with miticide number 2 next year. Come back to miticide number 1 after a full field season. Resistance in spider mites is somewhat unstable. All right, as the dry weather progressed, we set fields up for earworm issues. And we did have quite a bit of earworm problems in southwest Delaware this year. Earworms were most severe in non-irrigated fields, fields that had dry corners, and double crop fields. These are charts of our pheromone and blacklight trap capture for corn earworm per night. This is Georgetown. We peaked at about 15 moths a night. Decent, but kind of low. Look at Laurel. We peaked at 45 earworms a night. That's the southwest corner of our state. New Jersey experienced the highest corn earworm pheromone trap capture that they've seen since they began monitoring their state in 2003. We had a cooperator maintain a pheromone trap for us over at Lewis on the other side of the bay. And our population at Lewis location was pretty similar to Laurel. New Jersey's earworm populations were 3 times higher. So they were averaging about 150 moths a night in their earworm traps at their peak. And where that's important is with the insecticides options that we have for earworms. You all have seen these data before. Virginia Tech has been evaluating corn earworm susceptibility to pyrethroids going back to 2003 using these treated vials. They'll put a moth in, and after 24 hours, they'll evaluate that moth for is it alive, dead, can it fly? Starting in 2008, they started seeing an elevated percentage of moths survive after 24 hours. Last year, they had 37% survivorship. This year, they were down to 26%, but it's still there. Now, Sally Taylor was kind enough to supply me with some of these treated vials, and we ran 137 moths in our vial tests. Overall, we had about 40% survivorship. At a bump up in September, but where most of our earworms that were causing problems were in August, in this mid-30s percentage. So my point on this one is our population earworm susceptibility to pyrethroids is very similar to our southern neighbors. I also want to make mention they will test hundreds and hundreds of moths. I tested 130. And that is a testament to the earworm population differences as you go farther south. A few folks asked me, when should I get concerned about earworms in soybeans? If you Google search corn earworm threshold calculator, the first result that you'll have is a website maintained by NC State where you can enter in your sampling method, your control cost, price of beans, and your row width, and it'll calculate the economic injury level. So using some pretty ballpark figures for corn earworm on narrow rows, we're looking at 2 earworms in a 15 sweep sample. So even with that decline in pyrethroid efficacy, if we can knock them below 2 in a 15 sweep sample, we're doing pretty good. I think if you get more than probably 2 or 3 times above threshold, that's when we start worrying about how well a pyrethroid might do on those earworms. Having said that, always scout a field after a pyrethroid. They are no longer providing us the control they once did. Scout a field before, scout a field after. And I'm going to get to that scouting the field after in a moment. We also had a sweet corn trial on station. And my original goal was to float a spray schedule based on pheromone trap capture and compare that to a fixed spray schedule. Our pheromone traps, starting around the second week of August, started going up to that 15 per night or more, and they stayed that way until we harvested our sweet corn. So what we ended up doing is we compared a spray schedule of 3 days versus 2 days, straight pyrethroid. The reason I did that is some states will adjust their pheromone trap capture based on temperature to get down in that 2-day range. We also had in Besiege and Warrior. So we sprayed Besiege, the first 3 apps were Besiege, the last apps were Warrior on a 3-day schedule. We had an 11-point difference in the percentage of perfectly clean ears between our Warrior 2-day and our Warrior 3-day sprays. Now these red bars, those are tip damaged ears, where earworm damage did not go more than an inch below the tip of the ear. Those can be chopped out in processing corn. When you factor that in, we didn't have any difference in our treatments. But for the clean ears, that diamide really helped. So if we just look at our spray schedules here, 3-day spray schedule, the diamide really helped. And the question that I have going forward is, can we stretch out our spray schedule a little bit if we have a diamide in there? Something I don't recommend because we don't have data on, but it's something I want to look at. Had a couple of folks call me about late season corn earworm management for field corn. Because of all the rain we had in the spring, we had fields that were planted in June into field corn. They were silking right when we had our peak earworm flight. So on the research station, we had a corn that has the Cry1AB and Cry1F in it and also a really good rootworm package. And so we decided to spray it. We sprayed it with Besiege at 5 days after silk. Now, the folks that were calling me, they were seeing earworms in the ears already. If we could kill those earworms and rescue that, we wouldn't have to spray sweet corn more than once. And that's not the case. But give the folks that gave me a call a really good visual. We dissected those ears about 3 weeks after we sprayed them. Even though we have a slight reduction in the amount of earworms in those ears, a single spray is not going to clean them up. I also wanted to mention the BT traits. This is a slide from Galen Dively looking at Obsession 2 sweet corn and its non-BT isoline. The non-BT isoline are these black triangles, and the red are the Obsession 2 expressing sweet corns. Galen Dively has been spearheading a Sentinel program looking at earworm and BT trait technology efficacy. You can see back in 2010, 2012, the BT technology was keeping our sweet corn ears pretty clean. This is percentage of damaged ears. But over time, efficacy has been eroding for corn earworm. The field corn that we had planted is Cry1AB and Cry1F, neither of which ever had good earworm efficacy. This field corn also had a really good rootworm package. It had Cry3A and Cry3435. That's a really good rootworm package. We don't have rootworms in Delaware, in Sussex County, not a whole lot of them. Certainly not on the research station. So I've got here printed off and also on the back table a copy of the handy BT trait table. Some of y'all may have already seen this. What this table does on the back side, it's got all of the trait packages listed in field corn with their BT protein and the insect pests that are effective on that. That's something I think is worth looking at when you're advising growers on variety selection for the spring. Now, I said that this trait was never very effective on earworm. What's its value? All of our BT field corn has a LEP trait in it. Not all of them are created equal, but they are all very effective on European corn borer. That is the primary target of our BT technology when BT corn was first introduced. And because of BT corn, European corn borer populations are suppressed regionally. It's very hard to get a lot of corn borer pressure, and I don't know of any entomologists that have been doing a lot of corn borer work recently because they can't get the pressure. It's still out there. We still get it in our black lights. It still causes issues in areas that don't have a whole lot of field corn. So these traits are still valuable. All right, moving on to sorghum. This past year we did not have a whole lot of sorghum in Delaware. We usually don't. This year we had even less than our usual low amount of acreage. But we had a field in southwest Delaware that had spots that went above earworm threshold. Earworms will cause about 5% yield loss with 1 earworm per head average. Using some ballpark figures, assuming a yield of 80 bushels an acre and $3.15 a bushel, 1 earworm a head is going to cause about $12.60 in loss on an acre. We had a couple of spots in this field that exceeded that. This is a sorghum head without any grain left because of earworms, and a little bit of grain left. So that's what earworm damage will look like in sorghum. Throughout the southeast, sorghum acreage has really taken it on the chin, and it's primarily because of aphids. We have a couple of species of aphids that get into sorghum in Delaware. The yellow sugarcane aphid is present throughout the whole season. It's a very large aphid. It has a very hairy body. Last year, for y'all who were here when Sally Taylor was talking about insect pests, They had white sugarcane aphids show up on the eastern shore of Virginia in sorghum. No one was looking for it, and it caused severe damage to those sorghum fields. This year we examined 4 sorghum fields for white sugarcane aphid. We found them only in 2 fields in really low numbers and late. Is this going to be a significant problem for us? We're really far north. White sugarcane aphid overwinters in the Gulf. It's still an evolving picture. Because we have yellow sugarcane aphid move in low numbers throughout the season, we also bring in a lot of our generalist aphid predators. So the bottom line with sorghum, it's not a plant and forget crop, particularly when the flowering to milk stage corresponds to our peak earworm flight activity. Be on the lookout for aphids. If we have a damaging population of white sugarcane aphid in a field, it's going to be really obvious. Those aphids reproduce very quickly. They produce copious amounts of honeydew. You can flip over probably any leaf in a heavily infested field and just see hundreds of aphids underneath of it. That's not the case for us yet. And I don't think it will be the case moving forward, but it's something worth keeping an eye for. All right, so I mentioned pyrethroids and the importance of scouting a field after a pyrethroid has been applied. First, to check for efficacy for earworms, and second, because pyrethroids will ding our natural enemies. That sets a field up for soybean looper. This past year, Virginia and North Carolina both experienced some pretty heavy soybean looper pressure. For us in Delaware, it's more of a spotty pest. It tends to show up in spots in fields. This here is a field in Carville. This field I was using for a mite trial. I said my mite trial was a complete bust because of entomopathogenic fungi going after the mites. So I stopped looking at the field and was looking at other fields trying to find a good looper population. Where did the loopers show up? Right underneath my nose when I stopped looking for them in this field. So Sally was generous enough to lend me some data slides from their looper trials in 2017 and 2018. A couple of things I want to point out. In 2018, they had sustained looper pressure. This here is 23 days after they start going out with their sprays. They're still getting quite a bit of loopers in their plots. Second thing I want to point out, our diamides in Virginia are not as reliable as they once were for soybean looper. They do have confirmed diamide resistance in Virginia. Having said that, let's see here. There's Prevathon at 14 and 20 ounces. Here's Besiege at 10 ounces. In 2018, here's Besiege at 8. And we don't have an equivalent Prevathon rate, but the diamides look like they're working a little bit better in 2018 than they did in 2017, but that's not reliable. Virginia got very good efficacy from Radiant, from Steward, and from Intrepid Edge. North Carolina, Dominic Reisig put Intrepid Edge out in his plots, and that is the only product that they recommend for soybean looper in North Carolina. Other thing I want to point out here is denim right here. This is currently not labeled for soybeans. The active ingredient is a very good worm product. It's emamectin benzoate. And then finally the rain came back and we all cheered, right? And then it kept on coming and we were like, when does it end? But the rain really did help us with our late season worms. Our late season worms are susceptible to entomopathogenic fungi. These worms are not quite full grown yet. This was a field that had a lot of fungal infected worms. Soybean looper will eat 80% of all the leaf area it's going to eat in the last 3 days of its larval development. Same with corn earworm. It's going to consume the most soybean seeds and clip the most pods in the last 3 days of its larval development. These guys got taken out well before that. So that was a good find. This field could have had a lot more issues than it did. So, summary for soybeans. Scouting is really key, especially after pyrethroid use, because that does set us up for late season defoliators moving in from the south, like Soybean Looper. Really good coverage is really important, especially since we are seeing an erosion in pyrethroid efficacy. And proper worm identification is really important. I do have a couple of soybean loopers here, a couple of live specimens and petri dishes. Soybean loopers, as you know, have 2 prolegs on their abdomen. Green cloverworms have 3. Green cloverworms you can kill by looking at them. Soybean loopers are really hard to kill. I've also got, I'm going to throw this up. I think most of my copies have been taken. There's a few of these left on the back table, I think. This here is a survey of insect pest losses in soybeans for Delaware 2017. I used some feedback from consultants, chemical field men, and tech reps to conduct this in conjunction with other states. The mid-southern states have been doing these surveys for the last several years. This is something I plan on doing again for 2018 to get really good estimates and decent numbers on how much our insect pests influence our crop. I need y'all's input. So I know that now that we're getting our soybeans out of the ground and getting our corn out of the ground, this is the time of the year that everyone is about to get surveyed to death. I want to be first so you don't have survey fatigue when I come asking. So I've got up here a copy of the soybean insect survey they use in the South. Feel free to take this if you want. I've got my contact information on this. Feel free to email it back or give me a call or anything like that. One thing I do want to point out about 2017, from what I was hearing from folks, stink bugs played a significant role in pest management. 2018, I haven't heard anyone have much of an issue with stink bugs. Also haven't heard a whole lot of issues going on with Dectes this year. Have any of y'all? I see a couple of heads shaking and a couple of heads nodding. In general, the picture that I've been getting is that our Dectes pressure overall has been lower this year. I think that our early season rains has helped that. Something that we did look at last year, or this previous season, was Dectes and sunflowers. Sunflower is an alternative host for Dectes. Dectes really like it. I planted a couple of sunflower plants in my backyard and Dectes showed up. Where'd you guys come from? Middle of development land in Sussex County. We sampled a few fields that had sunflowers planted nearby. And we split the sunflowers and we split the soybeans. In Bridgeville, 17 of our 47 sunflower stems had confirmed Dectes in them, versus 2 out of 25 soybean plants in an adjacent field. And I say confirmed Dectes for sunflower. There are other insects that will get into sunflowers. Those are only stems that we actually dug out at Dectes. In Georgetown, 8 out of 50 sunflower stems had confirmed Dectes versus 3 out of the nearby soybeans. In Harbison, another weedy host for Dectes is cocklebur. 5 out of 18 cocklebur plants had Dectes in them versus 29 out of 1,600 soybean plants in the adjacent field. I think that we can use this to help manage Dectes. The folks out in the Midwest, out in Kansas and Nebraska, also deal with Dectes. They also have commercial sunflower acres. They think that sunflower can be used as a trap crop to keep Dectes out of soybeans. And I think that it's a good possibility. So if any of y'all have growers that you consult for or that you know who put in sunflower plots, I would appreciate the opportunity to look at these sunflower plots in July, to look at Dectes on the sunflowers, Dectes in the field, and then again either before or after dove hunting season to sample those stems. But I think this is a potential way we can manage Dectes. A couple of other things, insecticide update. Right now, the EPA is appealing the Ninth Circuit Court panel decision on Loy's ban. We expect this is going to take some time to work through the bureaucracy, so we fully expect that Loy's ban will be available through the 2019 season. Torac, active ingredient is tolfenpuride, now has supplemental labels for brassicas, fruiting vegetables, cucurbits, and potatoes. It's a broad-spectrum, contact-only insecticide from Nichino. Vidate, EPA is proposing usage restrictions on Vidate, lengthening reentry intervals for various crops, lowering the usage rates per acre, and removing a few crops from its label. And BASF's Insculus, or Phytopyropen, has now been labeled. This is a translaminar aphid product for soybeans, vegetable fruit, and tree fruit. Finally, have you all been hearing of spotted lanternfly? Yes? No? Don't know what it looks like? I have a couple of samples in alcohol. It's a very distinctive insect that is consuming Penn State's extension. We've been picking up, DDA has been picking up spotted lanternfly here and there in Newcastle County. This insect is causing severe damage to grapes in Pennsylvania. It has the potential to cause significant issues in orchard crops. It has been reported to feed on 70 plus hosts. If you see this insect, you can report it at this email address to DDA. And like I said, I do have spotted lanternfly preserved with me in case you want to take a look at it. Like I said, it's very distinctive. We don't have any other native species of insect that looks like it. And with that, I need to make a couple of acknowledgments. First to Bill Sissel and the poor folks that decided to work with me and didn't know what they were getting into. Also to Sally Taylor and Galen Dively for allowing me to bounce off harebrained questions off of them during the season and for sharing data with me. The Carville Farm Crew, Delaware Soybean Board, Sussex County, and our agrochemical industry partners who really help us do our jobs and give us support, and also y'all. I rely on your feedback and your observations, and I appreciate the insight and the wisdom that you have spread out in footprints over more fields than I can see. So I appreciate that, and I'll take any questions that you may have. The most work that's been done with planting green is from Penn State. John Tooker up there is the entomologist. Slugs will feed on cereal grains before they feed on soybeans, but they do like legumes. His idea is, can we leave some of our cover crop to give those slugs something else to feed on while our crop is coming out of the ground? His data looks promising. There are other agronomic issues with planting green. And those stalks will also conserve some moisture. So slugs will sometimes kind of stay near that, but that's just a field observation. Can it help? I think it can. But we need to make sure that we have our other agronomic practices lined up to make it successful. One thing that Penn State will stress with planting green is we need to be scouting for other insect pests like cutworms.